This paper is perhaps one main reason for which you asked me to give this lecture. Published in 2000, it was the first demonstration that you could assemble a vector, in this case a lentiviral vector, encoding the right composition of enhancers, introns, promoters, chromatin regulators that would allow to express enough beta-globin to treat a mouse with beta thalassemia. As you said, it took tenacity to get to that point. And for the students and postdocs who are listening today and who do undertake ambitious projects, they should know, as you already pointed out, Steve, that tenacity is key, and it took 11 years. It took me 11 years to get to this vector. The first 5 as a postdoc at the Whitehead Institute, and the next 6 when I opened my lab at Memorial Sloan Kettering. I would like to dedicate this lecture to some people. And first, Farid Boulad and Isabelle Rivière. They are my long-term collaborators on this big adventure, really. Dr. Boulad had transplanted many patients with thalassemia and sickle cell disease before I met him, and has been the PI on all of our Globin-related clinical trials. And Dr. Rivière established a stunning operation here to enable cell and gene therapy at MSK, starting from a staff of 1, and today approaching 40, she has put into place a facility that has produced CAR T cells for over 300 patients, expanded cord blood cells, of course, conducted the thalassemia trial that I will be discussing in a few minutes, has also produced DNA vaccines for melanoma, and even dopaminergic neurons derived from ES cells that are soon going to go to the clinic at MSK. I would like to also dedicate this lecture to Lucio Luzzatto, who was then my chairman several years back, who had joined MSK coming in from the Hammersmith, like you, Steve, in London. And he supported this effort in developing a gene therapy for thalassemia in the days where this was not so popular. People thought gene therapy would be dangerous, that recombinant viruses were to be avoided. And even in the community of hematologists, there was a strong resistance to this concept. And Lucio was instrumental in enabling us to build a facility that later made our trial in thalassemia and our CAR T cell studies possible. I also want to dedicate this lecture to 3 of our clinical collaborators who are taking care of our thalassemia patients. Aurelio Maggio from Palermo, and Renzo Galanello, now deceased, and Paolo Moy from Cagliari. I also want to dedicate this lecture to George Stamatoyannopoulos. Steve, you reminded us of his vision when he established this society. And he's one of the great geneticists and hematologists of all times. As you will hear, I will refer very specifically to 2 of his contributions, amongst many more, in the course of this talk. And lastly, I want to dedicate this talk to the patients that have participated in the trials that we have performed. The first 5 patients were in a trial to mobilize hematopoietic stem progenitor cells in adults with beta thalassemia. The next are 4 patients whom we treated on our early beta thalassemia gene transfer protocol. And then another 18 patients with sickle cell disease who generously participated in the first trial that we ran here at MSK to demonstrate the safety of using plerixafor to mobilize hematopoietic progenitor cells in patients with sickle cell disease. Here are my financial disclosures. There are none. My institution has dispensed with our intellectual property free of charge. So The Thalassemias and also sickle cell disease are related in many ways. And first and foremost, they affect the same gene, the beta-Globin gene, a little gene located on a human chromosome 11. Only 3 exons, 2 introns, from the promoter to the 3' enhancer located after the polyadenylation signal. You could fit the Globin gene in about 3 kilobase. And yet this gene is the source of 2 of the most common monogenic blood disorders on our planets. First are the beta thalassemias, which you see on the left part of this slide, for which today 449 different mutations have been identified. Most of these mutations affect the beta-Globin gene itself, but some are located far upstream, defining what has been called the Spanish or Dutch thalassemias, which I'll mention later. And to the right, sickle cell disease, which is accounted for by only 1 mutation in a codon 6, that changes the properties of the hemoglobin protein. In a very rapid way, the pathophysiology of these 2 anemias could be summarized like this. On the left here is beta thalassemia. It is a deficit. There's insufficient expression of the beta chain of hemoglobin. And this creates an imbalance between the alpha chain production and beta chain production, which impairs then erythropoiesis and leads to hemolysis. And this therefore results in an anemia, which can be treated with lifelong transfusions, not just occasional transfusions, but patients after the 80s, which Steve just told us about a few minutes ago, went on to programs of systematic transfusions every 2 to 3 weeks. These are life-saving, but they too lead to a major complication owing to iron overload. Iron is hyperabsorbed from the gut, but also provided through these repeated transfusions, leading to secondary deposition of iron in many organs. And in fact, cardiomyopathy, if without further treatment, is the major cause of death in transfused patients. Now, of course, there are iron chelators today that, if the patient is compliant, can limit this otherwise lethal iron overload. On the right is sickle cell disease. And in this case, the level of production of the beta chain is normal, but the beta chain bears a mutation. And this leads to the polymerization of hemoglobin in red blood cells. And I think you all know that under those circumstances, they become rigid and acquire sickle shape and can cause vasoocclusion and pain. And this can lead to strokes, for example, and can be lethal. So 2 diseases that affect the same gene, but both leading to anemias, but with systemic consequences, but really a different underlying mechanism. I just received a few days ago on May 8th, the traditional annual press release from the Thalassemia International Federation. May 8 is not just the end of World War II, it's also Thalassemia Day. And in many countries around the world, in Greece, Italy, India, and others, there are events to celebrate and plead for more research for this disease. The picture on the left here depicts patients that we shouldn't see anymore today. You see all the hallmarks of thalassemia in this untreated child: the hepatosplenomegaly, the large abdomen. You see the abnormal nasal bridge, the malocclusion, all this because her bone marrow is desperately trying to make more red blood cells, which of course cannot be achieved given the mutations of the beta-Globin gene. I excerpted this part below that you see here from this press release from just a few days ago, which says that patients with thalassemia still do not surpass the age of 20 years old worldwide. Of course, again, this is not the case in North America or in Europe and some other parts of the world where patients have access to thalassemia and chelating agents. But the burden of this disease in many countries remain very major. Another point of background I want to make, referring here to Janet Watson, a physician actually in New York, in Brooklyn, who in 1948 published a paper analyzing over 400 children with sickle cell disease. And she noticed that there were no sickling properties in the blood of infants for the first 6 months of life, and speculated at the time that the early hemoglobin found in these infants was different from the sickling hemoglobin that they would express later on and cause their disease. And indeed, today we, of course, know so much about the switching phenomenon, moving from the embryonic to fetal globin gene expression during fetal life, and then turning on this beta-Globin gene after birth, but which, as you see on this curve, still has some residual gamma-globin expression after birth, and more so in some individuals. And those individuals who have a genetically encoded persistence of the gamma-globin gene fare better when they have either thalassemia or sickle cell disease. And this collectively has been referred to as the hereditary persistence of fetal hemoglobin. And human geneticists have been studying this for many years, and I shall come back to this later in this lecture to tell you about how it not only informs our understanding of the disease, but leads also to new gene therapies based on gene editing. So going back over to 2-3 decades ago, the reasoning was the basis for the treatment of beta thalassemia is a cell therapy. It's this red cell transfusion that you give every few weeks. These transfusions are life-saving, but still carry a lot of risks, not just the iron overload, also HIV at some point, other infections, sometimes allergic reactions. And some patients actually are so immunized that they could not tolerate any transfusions anymore. And so you could cure these patients with allogeneic bone marrow transplantation by transplanting hematopoietic stem cells with normal globin genes. And you could do so safely if the patient was fortunate enough to have an HLA matched sibling donor. However, a majority of patients do not have an HLA matched sibling donor in thalassemia, and even fewer do in the case of sickle cell disease, which really provided a fundamental rationale for a genetic approach to treating these disorders. Now, if you wanted to introduce a functional globin gene in the hematopoietic stem cells of these patients, these are the properties you would want to reach. You would want the expression to be lineage restricted and erythroid specific. So that's different from all the vectors that you are familiar with that have been used for the treatment of severe combined immunodeficiencies or metabolic disorders, where typically non-specific promoters have been used. You would want this expression to be differentiation stage specific, initiating as the cells mature towards the red blood cell stage. Critically, expression has to be elevated to be therapeutic and to fill up these red blood cells, in the case of thalassemia, with enough of the beta chain of hemoglobin to extend the life of these red blood cells. And you would want the expression to be elevated also for safety reasons, such that you could be therapeutic following the introduction of only 1 or 2 copies of the vector per cell, not 3, 4, 5, or 10, which at least some of us would deem to be a concern. So this high expression is indeed critical, and you would want the last bullet, of course, expression to be sustained over time. Below here on the right, I give you a metric for the level of expression of beta-globin that would be needed in comparison to, for example, ADA deficiency, where you see there's perhaps 2 × 10⁻³ picograms of that enzyme per cell, more protein in the case of Wiskott-Aldrich. But the beta chain, as you see, is a logs above what is sufficient for a number of enzyme deficiencies. And indeed, the beta-Globin gene, together with some muscle genes or albumin, is one of the highest expressed in the body. So I won't go through the early pioneering efforts that some had tried 30 years ago. The work of Mark Rudin, of Richard Mulligan, and a few others. Those brave efforts failed. They could not express enough of the beta-Globin gene in murine chimera, bone marrow chimeras. And so, as Steve already mentioned, it's really this groundbreaking finding by Frank Grossfeld that revealed to everyone that we should not be only looking for enhancers, in the vicinity of the beta-Globin gene, and that some critical control elements could be located far upstream from the gene. There had been early insights by Dorothy Twan about this, but it's Frank Grossfeld who performed this critical experiment on the left, where he took this chunk of DNA that you see here on the left, encompassing these areas known as DNase 1 hypersensitive sites that you see here that I'll come back to, and juxtapose them to the beta-Globin gene, put another hypersensitive site that he found downstream of the beta-Globin gene, and in doing so was able to achieve high-level expression of the beta-Globin gene in transgenic mice. The beta-Globin locus control region turns out to be a master orchestrator of what happens to all of the Globin genes on chromosome 11. And as you see in the right here, these disparate elements, in fact, come together in the three-dimensional space in the nucleus, forming what Peter Fraser called chromatin hubs, where these DNase 1 hypersensitive sites that really map enhancers or enhancer-like elements come together, and where there is a high concentration of transcription factors and DNA polymerase. Now, having this understanding, as Steve already said, didn't mean that one could immediately construct a vector. These elements were far too big. And furthermore, they were genomic elements, not the cDNA or promoter that you typically introduce into constructs that you may use for whatever purpose. This is genomic DNA, which really destabilized the RNA viruses, the retroviruses, the gamma retroviruses that we were using at the time. In fact, some people lost confidence in this approach, and many abandoned their work on beta-Globin gene therapy. Some turned to using adeno-associated virus, others explored alpha viruses, but we were immediately interested in the lentiviral vector because of its rev-responsive element, and thought that perhaps the presence of the sequence could mitigate the instability and splicing that we had found occurs commonly in these complex vectors. And in 2000, we reported this study in mice with thalassemia. These mice have 8 to 9 grams per deciliter of hemoglobin, and you can treat them with this vector encoding locus control regions, elements HS2, 3, and 4, driving here the human beta-Globin gene, and you can treat their anemia. We had, as I mentioned before, this was the result of 11 years of investigation. We had other vectors that by 1998, 99 that could treat TNS 5, 6, 7, 8, 9, but TNS 9 in our hands was the best, and that's the one we published. In the next years, we showed that this approach of lentiviral-mediated globin gene transfer could correct the anemia, prevents secondary organ damage in these mice that even though they are not transfused, they too accumulate iron in their spleen, in their liver, their pancreas, in their heart. And you could see an alleviation of that. We could achieve long-term expression, which you see here on the right, by transplanting cells from primary mouse to a secondary mouse to a tertiary mouse, and consistently see the formation of this chimeric hemoglobin, which was made up of the vector-encoded human beta chain and the murine alpha chain. We showed that, as you would expect, this confers a selective advantage in peripheral cells, in red blood cells, owing to their normalized, or at least improved, alpha to beta globin chain ratio, and also that this was vector copy number dependent. In fact, a whole genetic pharmacology, if you like, was created of these vectors, and I'll show you an example of that here. And so on the y-axis here is a metric for the if you like, the potency of these vectors, which in this case is the gain in hemoglobin that you see in thalassemic mice. So if their baseline is at 8.5 grams per deciliter, they may go up to 10, 11, 12, or 13. And this is the improvement. And it's measured 6 months after transplant. And on the x-axis here, you see the vector copy number measured in the peripheral blood of those mice. And we purposefully do not want a high vector copy number so that most cells harbor essentially 1 copy per cell. And you can see that some vectors are better than others. Each dot here is a mouse, and you can see they form these little clouds, speaking to the relative position independence of these vectors, since the level is the same from one mouse to the next. And of course, therefore, in different hematopoietic stem cells. And some vectors are better than others. We strive to have the same vector copy number in all mice. It's not always possible, but you can see that some vectors, despite a higher copy number, wouldn't express as much. And so this shows how you can really fine-tune I would call it the pharmacology of these vectors. Now, after we published the work on TNS9 in 2000, several other groups turned their attention to lentiviral vectors and reported on different Globin vectors that overall have the same structure. They have a Globin gene, or sometimes a gamma-globin gene, in reverse orientation, because this gene you must use the gene and retain the introns. The beta-globin cDNA is not a good template for making protein, and using HS2, 3, and 4. What you'll note here is that if you do that metric of how much globin do you get per vector copy in a mouse 6 months post-transplant, you'll see that some of these vectors produce more than others. And therefore, while they are similar in structure, the details matter. Before moving, I just wanted to mention that Globin genes are everywhere, and you'll see what I mean by that. So much is known about the Globin genes. One of the first genes to be cloned, the gene involved in thalassemia and sickle cell disease. And many first studies in biology have been reached through the study of Globin genes, whether they deal with polyase or introns or ribosomes or the capping of the transcript, etc. And I want to just show you examples here. Some of you will recognize these constructs called PMDG or PMD-GAGPOL. And these were vectors constructed to express the VSVG, which back in the 80s, in the mid-80s, was not so easy to express. And so when Dan Ori came to see me asking for some advice, I told him you could use the beta-globin gene, take advantage of its second intron and polynylation, and sure enough, express the VSV gene cells, which otherwise, at least at the time, was difficult. And many derivatives of this were made. And if some of you wonder where the name PMD comes from, well, he named it. And so it's Michel and Dan, and that's the family of the PMD constructs. Another example of Globin genes outside of the Globin world, another study of our own some years ago, in 2008, you see, where we demonstrated that we could express therapeutic levels of factor IX, specifically from red blood cells in hemophilia mice that had been transplanted with a vector similar to TNS9, but encoding factor IX. When RNAi came along, we also explored whether the Globin gene could be harnessed to help the expression of RNAi and confer specificity upon it. And in this study, we showed that we could actually introduce these short hairpins in the introns of the globin gene, and that when these introns would be spliced from the lariat that was formed, an active small interfering RNA could be formed, which in this case knocked down the mutated sickle RNA, while the vector itself expressed a therapeutic globin gene. And the last example here is actually not from our lab, but I could not resist just mentioning it. And it's in the mRNAs that we're talking about so much today, and especially at this meeting, in the context of either cancer vaccines or, of course, COVID. A number of these actually utilize either a capsid or a poly-A that is derived from either a beta-globin or an alpha-globin gene. So back to our story. So with TNS9 published, and despite the quite widespread opposition to gene therapy in those years, in the early 2000s, we decided to move forward and had to present our project to the RAC committee at the NIH in Bethesda. And we had a public review which endorsed our protocol. It endorsed the protocol, however, pointing out that we should not enroll children, and also that we should not use a myeloablative conditioning regimen. And so we did. We also followed upon the recommendation to conduct a stem cell mobilization trial, for it wasn't sure that the GCSF mobilization procedure would be safe in adults with beta thalassemia. And we conducted that trial, and to this date, GCSF mobilization is used, I believe, in pretty well all thalassemia trials. And my colleague, Isabelle Rivière, and her team with Xian Wang and others divide this protocol, which is one that all of you who work with hematopoietic stem cell gene therapy are familiar with, collecting these, the CD34 positive cells, setting up a backup, a cryopreserved backup, then transducing them with the vector, and cryopreserving these cells, during which time much release testing is performed, a vector copy number, but of course the detection of RCL, and then infusion after conditioning, which again, in this case, would be a reduced intensity conditioning. Now, I want to pause there and say that others were starting trials in this space. There had been a first trial in Paris that I'll come to in a minute, where 2 patients were treated. We treated the first in the US and the third patient with beta thalassemia. But 2 other trials were in preparation at the same time. One that makes use of a vector called BB305, which was sponsored by Bluebird Bio, and another one called Globe that came out of the beautiful work of Giuliana Ferrari in Milano. Now, in that first trial in Paris, in the second patient that was infused, a serious adverse event took place. It took place in the form of this expansion of a single clone, which you see here expanding over time, becoming more than 1% of all circulating nucleated cells harboring a vector in the blood of this patient. The integration was found to have taken place in this HMGA2 gene, a gene involved in particular in some benign tumors, but in proliferation. And the integration in the intron, in the third intron of this gene, led to a high increase in the level of transcript and removal, owing to some splicing reaction occurring into some of the vector, the removal of the 3' end of this transcript, which happens to contain regulatory regions. Now, expression of this gene was vastly increased, 10,000-fold over normal levels in red blood cells. And that increase could not be accounted for solely by the deregulation of the transcript. There was still a 200-fold higher than normal induction of that gene that the authors of the study said could not explain. Now, a key question, that's in red cells, key question is what was happening in the very early cells that constituted this clone that went on for 7 or 8 years, I believe, and fortunately never acquired additional mutations and never became a leukemia. That they could not measure. But something had to have happened in those early progenitors. So that serious adverse event, thankfully, did not stop this line of investigation. And in fact, the trials went along. Bluebird Bio took over that vector, removed an insulator that was there in the origin, and that is the BB305 construct that those of you in the field know of today. And the Globe trial opened, and so did ours. And here's a summary of the reported patients from these trials today. Here's the vector copy number that were measured in the infusion products administered to these patients with beta thalassemia. Some are adults and some are children. I'm not going to break that down here. You can see that over time, the vector copy number is getting higher and higher. The more recent trials, you see copies of 1.9 to 5 or even up to 4. Now, the good news is that a majority of these patients have become transfusion independent. Of course, we need to see how this will hold up over time, but after 18, 24 months and more, many of these patients remain transfusion independent. So what happened to our trial? Well, sometimes you get unlucky for being first. And the way the vector was manufactured, which wasn't purified to the level that subsequent vectors were made, it turned out did not allow us to have as high gene transfer as we had hoped. And the level of gene transfer that we obtained in the 4 individuals that we treated was in the range of 0.2 to not quite 0.3. That's the average, the mean vector copy number in the population of CD34 cells that you infuse to the patient. Now, we had hoped in the beginning that that could be enough to achieve transfusion independence, but it is clear now, it became clear that that would not be enough. Having said that, we've seen some response in 2 of these patients. And as you see now, the long follow-up here at 96 months, some up to 7 or over 7 years of follow-up, you can see here their vector copy numbers hovering around essentially 0.05 or 5% of nucleated cells in the peripheral blood. And in 2 of these individuals shown in red here in subjects 2 and 4, you see actually a decrease in transfusion requirements, which of course is very nice to see, but not enough to achieve transfusion independence. Having said that, we can learn a lot from these patients, and we have been monitoring the integration sites over time. And now we have a study that's now 7 years out with a large body of integration sites that we've analyzed with the help of Rick Bushman. And so these patients obviously do not have any hematological adverse event. They're perfectly fine. But at a biological level, we observe clonal expansions. They're moderate clonal expansion. The biggest one is this one here in one of these 4 subjects, where you see starting at year 3, one of these clones that accounts for more than 5% of all nucleated cells, circulating nucleus cells, remaining here year 4, year 5, and at year 7 now, it's actually 17% of all peripheral blood cells. You wouldn't have seen that in these individuals, in any of these, at 18 months or 24 months. But as we follow over time, we do see this emergence. So why could that be? Well, the literature on lentiviral vector gene therapy, as you know, shows that lentiviral vectors are, by and large, very safe. I think we all agree that they all are much safer than the gamma retroviral vectors. Having said that, there are some events that can occur. I'll start with the CAR T cell fields. The disruption of TET2 or of a CBL-B has been shown. This is from gene disruption of genes with regulatory function to lead to clonal expansions. By and large, however, in long-term follow-up studies up to 10 years, there has not been a clear relationship between the site of integration, the proximity to a cancer-related gene, and the preferred expansion of those cells. On the other hand, though, in HIV patients, this is not gene therapy now, clearly some integrations have been associated with long-term persistence, with disruptions of genes such as BAH2 and others. In the case of hematopoietic stem cell gene therapy, well, obviously there's the case of the HMGA2 disruption that I just discussed before. And we have data, of course, data from the studies and outside of the SCID of the world of lymphoid deficiencies. We have, of course, studies in adrenoleukodystrophy or metachromatic leukodystrophy that so far have shown that, again, there are no hematological adverse events, certainly nothing that looks like a leukemia. No case of MDS either reported so far. Having said that, all these authors and these papers that I'm showing you here and more that I couldn't put on this crowded slide note integrations next to genes associated with either proliferation or apoptosis or so-called cancer-related genes in a very broad sense. So again, well tolerated, but I believe that this is something that we ought to continue to monitor carefully. Which brings me back to our Globin vectors. They are thought to be erythroid specific, and there's plenty of work has made that point before. But here I show you the profile of their accessibility, if you like, shown in the form of attack-seek peaks. The profile of those locus control region elements that I discussed before in different cells. So in CD34 positive cells, it's clear that HS1 and HS2 are accessible. There are factors binding there. In erythroid progenitors and megakaryocytic progenitors, these sites too are an open configuration, if you like. And HS3 and HS4 become so as well. Obviously, in erythroblasts, all four sites are well known to be open and busy with expressing the beta-globin gene in adults. In fact, if you look at other mature hematopoietic cell types, such as T cells, you see that even HS2 remains still open in this non-erythroid cell type. So it is possible, at least in principle, that these sites could adopt a structure in non-erythroid cells, which could be permissive or even conducive to transcribing neighboring genes outside of the erythroid lineage. And so we decided to test this. And so we introduced these elements that I'll show you in the next slide in hematopoietic stem cells in mouse, and introduced them at first with a minimal globin promoter and a GFP reporter. And with these FACS panels, looked for whether they had any activity in any of these early progenitor cell types. And so there's a range of these constructs. And here I just put up 4 of them with HS1, 2, 3, and 4 driving GFP. And these are the expression data seen in the bone marrow cells a few months after transplantation. And it is clear that HS1 and HS2 in particular drive expression of GFP in these mice. We see it in these long-term, short-term bone marrow cells. Of course, their function increases over time, and in particular, if you see below now, if you go to the erythroid lineage, now you see in the same animal, the full effect, if you like, of these enhancer elements, increasing GFP expression in the erythroid lineage. But as you see, they actually are active in early myeloid progenitor populations. In fact, this is something that George Stam and I had discussed for some time before we had these data are very recent, much more recent, what I just showed you and unpublished. But we had a suspicion that some of these HS elements might be active outside, again, of the erythroid lineage, and shared a common belief that insulators could perhaps restrain the function of these elements. And George undertook a very thorough exploration trying to capture all possible insulators in the human genome. And he, together with David Emery and John and Mingdong and others, reported in Nature Biotechnology this beautiful study where they identified a number of human elements with either barrier function or enhancer blocking function. Well before those were published, George shared the A1 insulator with us, which had a very good enhancer blocking activity. And so we tested in these mice. And so here are the Globin vectors shown above here. Here's the TNS9 kind of Globin vector, except that it drives expression of GFP that you see here, next to one that is flanked in the integrated form by the A1 insulator. And again, this is from bone marrow chimeras, and you see here the expression of GFP. It's low, but it's there in these various early progenitor cell populations in these mice. And indeed, when the construct is flanked by an insulator with enhancer blocking activity, such as A1, we see a reduction of this activity. Nicely, this does not end up reducing in any way the level of globin expression or GFP expression here as a surrogate in the erythroid lineage. But what it does is through insulations, presumably block interactions at the site of integration with neighboring elements that can lead to this leaky expression, if you like, in early progenitors. So the field of Globin vectors is still alive and well. Recently, 3 groups published new vectors. The group of Don Cohn, the group of John Tisdale, the group of Stefano Livella. You can see that all of them are still essentially in the same mold, a globin gene in reverse orientations with the LCR elements. We have our own too, called SNS23, which we think very importantly is flanked by an insulator, the A1 element, but also in which we've introduced a number of other mutations that, amongst other things, allow for much better gene transfer through mutations of elements that destabilize the transcript and packaging cells. And as you see here, without the addition of any transducing enhancer, we can now achieve levels of gene transfer that we simply could not with the earlier version of TNS9. And so at this point, we're poised to launch such a clinical study. Now, I want to address, before finishing, the advent of genome editing. While all this is going, many are making progress in identifying, zinc finger nucleases, talons, and of course, CRISPR-Cas9. And this is my opportunity to, like so many at this meeting, to again salute Emmanuel Charpentier and Jennifer Doudna for their wonderful work. And this obviously opens additional possibilities, targeted gene addition, targeted sequence disruption, or mutation repair. And here are some of the contributors below some of this work, but there are many more, and a lot of it is presented at this meeting. So let me make a comment here about BCL11A and come back to those hereditary forms of fetal globin persistence that I mentioned way in the beginning. Well, of course, the Greeks gave the name of thalassemia to thalassemia, but I think we owe it to the Sardinians, and in particular, Antonio Cao, to have collected the genomes and the pedigrees of families in Sardinia who had persistence of fetal hemoglobin, and who, when he teamed up with David Schlesinger in Hopkins, to do this big gene hunt and come up with BCL11A as one of the key regulators mediating the silencing of the gamma globin gene. Stuart Orkin then demonstrated the binding and how this repressor of fetal globin operates. Interestingly, and I said I would mention George another time, many years before, 1985, guess who, none other than George Stamatoyannopoulos, had reported this mutation in the promoter of the gamma globin gene, which, of course, he couldn't know at the time, but which disrupts the binding of BCL11A. So this now, the advent of all these gene editing tools now opens up a number of possibilities for the genetic treatment of the thalassemias and sickle cell disease. And you may either choose to induce the fetal hemoglobin by allowing for expression of the gamma globin gene, or on the left here, you may elect to restore or add to the expression of the adult form of the beta chain of hemoglobin. So let's quickly walk through this. So if you want to express more fetal hemoglobin, one approach is to have a lentiviral vector encoding the gamma globin gene, such as Poonam Malik has reported. Another way is to use a lentiviral vector coding for an shRNA that would downregulate BCL11A expression again, as shown by David Williams. But now with tools such as CRISPR-Cas9 or zinc fingers or base editing, you can attempt more things. For example, you could delete the delta and beta globin gene, recreating what is, again, the human geneticists described years ago as deletional HPFH. Another way is to mutate the enhancer, that the erythroid-specific enhancer that drives expression of BCL11A. Or you can target the gamma globin promoter itself. And in this case, either disrupt the binding sites for repressors or create binding sites of stronger activators. Now on the left, as if you wanted to use gene editing to restore the production of an adult hemoglobin. And you can use CRISPR-Cas9 or TALENs or zinc fingers to correct the sickle mutation. This is applicable, of course, to the sickle mutation, or avoid a double strand break, and perhaps by using either base editing or prime editing, reverse this infamous mutation. You can also use continue with gene addition. Obviously, the lentiviral vector is encoding the globin gene is what I discussed before, but you can also now target the delivery of a therapeutic beta globin gene, which I think is particularly relevant to the thalassemias, where there are over 400 different mutations, not one mutation like in sickle cell disease. And several authors have already reported the targeted delivery of the beta globin gene in either the hemoglobin gene itself or promoter or exon 1, or introducing the beta globin gene and the alpha globin gene locus, or targeting the ROSA 26, or putting it in the locus control region as Eric Bouhassira has shown, or to target extragenic safe harbors. What are those? We defined extragenic safe harbors as regions in the genomes where we think we could safely introduce foreign genes. And we created a list of criteria for this based on the extensive literature on insertional mutagenesis in hematopoietic stem cell gene therapy. And so with these 6 criteria that we see here, we create an atlas of the human genome that meets these criteria. But wanting to target the delivery of genes to those sites, we added more criteria. We want these sites, the ideal targeted sites, to be easily cleaved and accessible, and above all, one that would allow for the regulated and sustained function of the exogenous gene or genes. And we hypothesized that these would lie next to attack-seek peaks. We first conducted this work in T cells, and we are currently performing it in hematopoietic stem cells. But we have therefore identified, by overlaying these two atlases, a number of regions in the human genome that meet these 8 criteria. And here I would like to show you early studies of what happens when we introduce now a Globin gene, and in this case, it's the Globin GFP fusion gene, controlled by some LCR elements in a genomic safe harbor. And this is in human CD34 positive cells. Here you see the result obtained with the lentiviral vector. And here you see the expression of the same design of a vector, but targeted to this genomic safe harbor, where we actually don't see any expression of GFP at the BFU or CFU stages, but it does come on late and comes on just as well as the lentiviral encoded Globin gene. So I'm almost done, but I've only talked about the genes, and I should say a few things about, I believe, the cells. I want to add something about collecting the cells, and how to collect the cells, and how to infuse the cells. I mentioned GCSF mobilization in thalassemia before, but there was a literature that warned us that we should not use this approach in individuals with sickle cell disease. There have been fatal outcomes or multi-organ failures reported in sickle cell patients given GCSF for any reason. And so we needed another approach, and we thought we could try plerixafor, a mobilizing agent that is well known, but we were told that this would be too dangerous or could potentially be dangerous in patients with sickle cell disease because of the precedent of GCSF. But we thought this was important enough to try, and we didn't think that there would be these complications. And so we wrote a protocol that was supported by the Doris Duke Charitable Foundation. We wrote an IND for it that was reviewed, of course, by the FDA. And Sanofi was kind enough to provide us the drug free of charge. And we started this cell as a dose escalation trial, and this was well tolerated up to the standard dose. And we eventually published these data. As soon as we showed them at meetings, before they were published, we noted that other groups immediately adopted it. And although this hasn't really didn't get any thanks for it, I'm pleased that today everyone in the sickle cell field is resorting to a fairly safe plerixafor mobilization of HSCs in patients with sickle cell disease. Now, how to administer the cells is another very big question here. And I just want to point out, in our 4 patients in thalassemia, you may recall we used reduced intensity conditioning, which is in contrast to the other trials, the HEB and trial, the Globe trial, as well as the first trials that were recently reported using gene editing in 2 subjects with beta thalassemia. And so I just want to point out that we have not seen any case of vaso-occlusive disease, which is a known complication of a high-dose busulfan, which has been seen in some of these other settings. This difference here is not statistically significant, but it is well known in the field that myeloablation exposes to the risk of VOD. But I also want to highlight the time to engraftment, which you see here in our studies looking at just the platelets of being 14 to 25 days, and the much longer times to recovery in these patients when they received myeloablative conditioning. And transfusing platelets, while feasible, can also be sometimes complicated in patients with thalassemia. So I think we really need a way to give these cells in a safe way. And I just want to finish with this slide before concluding and highlight the advent of antibody-facilitated chimerism. And also, this gives me a chance to mention here my Ph.D. advisor, Thomas Wegman. Before I joined his lab, he was studying, amongst many esoteric topics, what happened to parent and F1 mice that were sewn together. And this led to what was called runting disease in some mice, and some mice died and others survived. Of course, we would call that graft versus host disease today. And he looked for why some mice did not die. These were parent and F1, like here, an A by B, like a BALB-C mouse and a BALB-C by C3H mouse. And found that there were antibodies, that in those mice that survived, there was an antibody to the B haplotype. So when in his lab, I reproduced this, by this time taking the bone marrow of mouse A and infusing it into mouse A by B, which doesn't give you any engraftment. But if you infuse an anti-B antibody, you see the chimerism. The mouse is converted to all donor, and this is without radiation or any chemotherapy. In fact, you can even infuse the cells and give the antibody later and see this antibody-facilitated chimerism take place. And I'm pleased to see today that targeting c-KIT, this was shown in monkeys by Thalassemias, and today, of course, we're all watching the studies of Judy Shizuru that perhaps will deliver a way to infuse these cells without chemotherapy conditioning. So this is my last slide, Steve. I find too many really lessons learned through all of this, whether it's vector design, trial design, safety, off-target activity, integration sites, regulatory aspects. To just summarize, I just want to say that I think that a genetic cure of the beta thalassemias and sickle cell disease, I believe, will be achieved. There are several technologies and strategies today, whether you want to induce a fetal-like hemoglobin or restore an adult-like hemoglobin, that are very exciting and appear to be very promising. Having said that, achieving high efficacy and safety, I believe, still remains to be demonstrated. And assuming that several of these wonderful approaches prove to be efficacious and safe, I do think, to be efficacious, I think the safety profile is perhaps ultimately the most critical differentiating factor of all of these, as well as their cost. I want to thank this time all the people who worked with me over the years. Above here are members of my lab who over the years worked on Globin gene vectors. These highlighted in 3 helped me with preparing the slides for today. I mentioned already my MSK collaborators and other advisors to this work as I presented this talk. My last word is to thank those who supported financially our work in order of contribution, first and foremost, the Stavros Niarchos Foundation, but also the NHLBI, Sloan Kettering Institute, Aaron Gene Therapeutics, the Doris Duke Foundation, the Leonardo Ciambone Foundation in Italy, and the Cooley's Anemia Foundation in the US. Thank you for your attention.